Okay, we're gonna get started.
Oh.
I got really bored last night.
That's her.
Okay. All right, the first thing you have here, this is an interpretive list. Second question, we have cartilage that's holding the ribs to the sternum. What kind of cartilage is that? It's called costal cartilage. Of the three kinds, it's called hyoid.
Yeah.
Here we have the spine and the hips. Each of these bones are coxal bones. There's one coxal bone, here's another. And the pubic part of the coxal bone, they come together, but they're held together by this cartilage, which also is good for resisting compression. Fiber cartilage. This is fiber cartilage. Pubic symphysis. You're looking at the, this is probably from a cow or something. Long bone, cut longitudinally. We're looking at the ends where you can see spongy bone. What kind of marrow do you find here at the epiphysis? Bone marrow. Then the question, we're looking at cartilage tissue. We know it's cartilage. How can we immediately say this is either cartilage or bone? What's the simplest thing to find?
There's no central canal.
Well, how can we? What I'm saying is, how do we know that this is cartilage or bone and not some connective tissue proper? It's all the lacuna. Yeah, you see all these cells and spaces. That's how we know we're looking at either cartilage or bone. We can rule out bone because we don't see either branches or osteons, those circular things. So some kind of cartilage. Looking at the extracellular matrix, how would you describe it? Give me some descriptive words. It's branched and dark. Which of the three has branched and dark fibers in the extracellular matrix? I can see it. Elastic. This is elastic cartilage. This is from where are major places where you find elastic cartilage? On the very tip of the nose, the ears. Most of your nose is actually hyaline. One other place. The epiglottis, which covers the larynx and trachea. Epiglottis. This is actually from the epiglottis. Cool. All right, that is our warm up. All right. Hey everybody. So what we're going to do is we're going to continue talking about bone. And then we will discuss, or I'll give you time to work on the next lab check as well as review the cadaver. There's cadaver work on, there's cadaver stuff on the practical next week. We have a practical on Monday. Yeah.
Yeah.
Practical on Monday.
Also, make sure you come with pants on.
Practical
And
pants on.
actually cover your skin when you go back on these.
Yeah.
So is lab check 4 due Wednesday?
Lab check 4. Lab check 4 will be due Wednesday. Lab check 4 will be due Wednesday. The rough draft, hopefully you did give me a rough draft because I'm going to give you feedback. But your final activity 3 will be due by the end of class. I'll give you your forms. I'll be grading it while you're working on your lab check. Or grading, editing. So I'll tell you what you need to change. And we'll get to that later. Are there any questions before we continue on bone? Questions? Okay. You left off on, oh yeah, our questions, and then also on slide 17. I want to show you the bone while back here. This is bone that's been treated in acid. This is bone that's been treated in acid. You have a bone that's treated with acid. Acid, like you hear about acid rain eroding cars and stuff, metal. So it erodes non-organic things. It erodes mineral things. We can't erode it. But if we have acid-treated bone, what is it affecting? Is it affecting the calcium more, or is it affecting the collagen more? It's affecting the calcium more. The bone is treated with acid. Here's an example of a bone that's probably a chicken bone that's been baked. You can see how it's more pliable. It can bend without snapping. That's what you got. It's not really like a but it's more pliable. So here's the best. All right. We left off on slide 17. We talked about the basic differences between. Compact bone. Compact bone is found towards the edges. You can see how much more compact it is in this bone versus spongy bone. Which looks spongy. Lots of pores. And you find spongy bone either towards the edges or towards the end, towards the epiphyses. While compact bone is just around the outside. Okay, so we know where osteocytes are found. Osteocytes is when they're within lacunae. They've surrounded themselves with matrix. If you look towards the very, very edges of bone, that's where you find osteoblasts. Just like what we saw with cartilage, chondrocytes are found towards the perichondrium. Here with bone, osteoblasts are found towards the periosteum. All right, if we're looking at the very outer portion of bone, so like this region here, you've got your spongy bone, that'll be right here. You've got compact bone, that'll be right here. And you've got your periosteum, which is the very, very outer layer. There are two layers of periosteum. The very, very outside of the bone is fibrous, it's dense, regular. The very, very outside is fibrous and protected, it's dense, regular, but towards the compact bone, which I'll color green, you've got layers of cells that have osteoblasts. So the periosteum here contains osteoblasts. So osteoblasts are found on this inner layer of the periosteum. They, of course, are what produce new matrix. The periosteum has osteoblasts, but there's one other place where you have osteoblasts, and that's the inner lining. It's not going to be a straight line because the bone's spongy, but lining the spongy bone on this side, there you can also find osteoblasts. You have osteoblasts in the periosteum towards the outside. You have osteoblasts on the inner lining. That's the endosteum. Endosteum. Because it's towards the inside of the bone. So it's lining the dewary cavity. So on either side, you've got osteoblasts, and in the middle, you've got osteocytes and matrix. You've got a sandwich of osteoblasts, osteocytes, and matrix. Here's an example of that. Whenever you have marrow space, and you see these cells lining that space, those are osteoblasts. If it's within a lacuna, it's an osteocyte, whether we're talking about compact bone or spongy bone. So if it's lining the marrow, it's an osteoblast. It'll eventually return to an osteocyte that builds that matrix. So two places where you find these osteoblasts, periosteum and osteum. There is one more bone cell type. There's osteocytes, osteoblasts. Osteoblasts mature into osteocytes. I said that when we build bone, not only do we have to add bone, but we also have to remove bone. It's important to do both. So osteoblasts help add bone, help add matrix. Osteoclasts remove matrix. We need a way to remodel, and this is how we do it. Osteoclasts, they're also found in that lining of the endosteum. Sorry, here. They're very big. So osteoblasts are just look normal size. If you see a really large cell facing the marrow lining, facing the cavity, that's an osteoclast. Osteoclasts are very big cells. How they get rid of bone is that they secrete acid. We just saw how acid can decalcify. So if these osteoclasts are secreting acid, you're going to remove bone. I know that sounds bad, but remember, two reasons why we want to remove bone. If we want to remodel something, like if we want to remodel this building, we have to tear some part of it down first before we can rebuild. So it's useful in that sense. And also, calcium phosphate is more useful. It's a priority to have calcium phosphate in cells like neurons and muscles versus bones. So if we're deficient in these nutrients, we'd rather use these nutrients for neurons and muscles than for bone. And so we can get it released into the bloodstream. One reason why these cells are so big is because they have more than one nucleus. So that makes them a big cell. This is, I guess, the first cell type where we've seen that cells have more than one nucleus. Yes?
Do all the multiple nucleuses share the same DNA?
Do the multiple nuclei share the same DNA? Every single cell in your body, minus your sperm or eggs, have the same exact DNA. It's more about what they express. So by having more DNA, what's the advantage? What's the advantage? If you have more DNA, what can you do with it? Create more. For what?
Create more mitosis.
You could you could undergo mitosis, but you can make more RNA. If you make more RNA, you can make more protein. Make more protein. So, can you think of another cell type that we haven't really focused on yet that has a lot of protein? When you think of protein, you think of this cell. Muscle. Muscle cell, skeletal muscle are multinucleate also. Skeletal muscles are multinucleate, so they can make. All right, your textbook goes into different types of growth in a lot of depth. We're just covering the surface here. So what I go over here is just the basics of how bone can grow. This word, appositional growth, is describing a direction of growth. Appositional is when, if you start with one size, narrow. So we go from one size to a bigger size, go wider. That would be appositional, going wider. Start with a narrow diameter, and over time it gets bigger. For this to happen, not only do we need to add more bone to these outer layers, so that it widens, but we also need to remove bone from the inner layers, because we want to widen the cavity. This is what I mean by remodeling. We need to add bone while removing bone when our bone grows. The term for adding bone is ossification. So these osteoblasts are ossifying to widen the bone. The term for removing bone is to resorb. I know it's a weird word. I don't know why it's not just absorb, but absorb is a technically different word. Resorb is to remove bone. Resorption. Ossification and resorption. So we need those two cells working together to allow for this type of growth. Questions on apposition? It's from the wire. If we want to go longer, the word for that is interstitial. We've seen that word before. Interstitial just means between. So this type of growth is because we're adding stuff in between the bone. Remember that I said that growth areas are in the metaphyses? If we want the bone to grow longer, we can add more bone to go in either direction. That's called interstitial growth. This region within the metaphysis where bone can grow, that region is known as the epiphyseal plate. I know it's in the metaphysis, not the epiphysis. It's in the metaphysis, and it's called the epiphyseal plate. This region has cartilage. Another region where you find hyaline cartilage is known as the epiphyseal plate. You can see the end of the bone here. Hope you can kind of see where I'm pointing. I know it's at a weird angle. But here's the end of the bone. This person, I can say with certainty, is young. They haven't fully matured. Because there's bone missing in these parts towards the end, here, 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 here. These are growth regions. Those are the epiphyseal plates. Regions where you have hyaline cartilage. That cartilage will be replaced by bone. So you'll add bone and cartilage, and you'll keep making the bone longer until a point where the whole thing gets replaced with bone. All the cartilage is gone. So osteoblasts are invading the areas where there's cartilage cells, which is chondrocytes. And it keeps growing until there's no more growth. No more cartilage, so there can be no more growth. Does that mean the epiphyseal plate is gone when they're fully grown? Yes. Good insight over there. See that? So there used to be cartilage here when this animal was younger, fused together, forms water. Direction of growth. These next two words are more about how the bone is laid down. This first type of ossification is called endochondral. Endochondral ossification. This type of ossification happens typically in your longer bones. So your limbs, your vertebrae, I know they're not long, but your hip bones also, which aren't quite long. But most of your limb bones are long bones. They have a longer shape. So this is a long bone. Endochondral, what that means is it's happening inside of what's chondro mean? Cartilage. It's happening within cartilage. Early development. Some of our bones or skeleton start out as cartilage. They're not really bones yet because it's just a cartilage framework. So really early development, when we're still fetuses, some of our bones, like our arm bones, they're not bones yet, they're just cartilage. And what happens is osteoblasts will move in towards the center of the bone. Osteoblasts will move into the middle. And if you have osteoblasts moving into the center, you can start replacing the chondrocytes. Chondrocytes get removed, chondrocytes start dying, and osteoblasts can grow and lay down bone. We form what's called a primary ossification center. Primary ossification center. It's the first place where this bone ossifies, the middle. Primary ossification center. Chondrocytes are dying, getting replaced by osteoblasts. Osteoblasts eventually mature to osteoclasts. osteocytes. The first type of bone to be laid down is spongy bone. So it's not yet compact bone. And while this is happening, you can see some appositional growth and interstitial growth happening. We're getting rid of bone in the middle with the help of osteoclasts, and we're adding more with the help of osteoblasts. Now that a cavity is formed here in this middle portion, that can house yellow bone marrow. What is this region of the bone called from here to here? The diaphysis. So we hollow out the diaphysis. That's where yellow bone marrow can fit. The first part of endochondral ossification. What about the ends of the bones? The ends are still. Now that this is hollowed out, we've got compact bone rearranging on the edges here. And I know we've skimmed over this. Like I said, I'm just going over the surface. Spongy bone is the first to be laid down, and then spongy bone can be reorganized into compact bone with the help of osteoblasts and osteoclasts. So we have compact bone, spongy bone, cavity. And now at the ends of the bone, at the epiphyses, osteoblasts migrate into the center, start ossifying and replacing cartilage. Eventually, you'll have all of this bone at either end. Now you're meeting in the middle here where there's still cartilage. Because you've ossified in the center, going this way, and you've ossified at the ends, going in either direction. So there's still a region at either end, a strip, where you still have cartilage. That's your epiphyseal plate, where you still have that region of cartilage. That's why it's there. Epiphyseal plate. And by the time we're born, we're still maturing. We still have these epiphyseal plates. That's known as growth plate. When we say growth plate, that's the epiphyseal plate. I want you to know the technical term, of course. Eventually, that gets replaced. You keep replacing chondrocytes here at this border. Osteoblasts move in, keep ossifying. And you eventually slowly replace it all until it comes to a line. And that's what's shown on that bone, that's what's shown here. Eventually that growth plate disappears, and now it's just a line. Epiphyseal plate, and at the ends here, in this area where you have spongy bone, that's where you have the red bone growing in between.
So then after that, the chondroblasts just die.
After that, the chondroblasts just die, yes. Chondroblasts die, you get these parts. Like I showed you on this x-ray, you can see it here too. When you're younger, this is the whole bone, but right now it's two distinct bones. Here's three. Here's one, there's a tip, and there's a tip over here. You can see lines that represent the epiphyseal plates, because cartilage doesn't show up on an x-ray, bones do. Once you're fully grown, that line disappears because the bone is fused together. So technically, you have more bones when you're younger than when you're older, because these bones are separate. They fuse together when you get older. That's one type of ossification. Endochondral. This one's a little bit easier. This is called intramembranous. With intramembranous ossification, you don't rely on cartilage. Intramembranous is where you start with so we just got a bunch of mesenchyme cells surrounded by a bunch of collagen. That's what cells in a sea of collagen. Some of them become osteoblasts. That's what mesenchyme cells differentiating into osteoblasts. Now that they're osteoblasts, they can start creating bone. They can start laying down calcium phosphate extracellular matrix. So here's some ossification happening. And once the osteoblasts build a wall around themselves, those calcified areas. So you still have mesenchyme in these surrounding areas, but now we've got an ossification area here, another ossification area here, and over here, just sporadic all over the place. There's no real order to it. And these keep growing. You still have an osteoblast to the outside. You keep ossifying. What will eventually happen is that this will fuse to here. This will fuse to here. This will fuse to here. What does that remind you of what we just discussed? What kind of structures do we form? I mean, how do we know? It's not compact. What do we call those branches of spongy bone? Trabeculae, yeah. We're forming trabeculae. They keep ossifying, they keep fusing together until we get this whole network of bone. That's what trabeculae are. Network of bone. That's all I want you to know about a network of bone. That's what trabeculae are. Network of bone. That's all I want you to know about intramembranous. Formation of bone, which form trabeculae. I forgot to mention, if endochondral ossification is most common in long bones, intramembranous ossification is most common in flatter bones. So bones of your skull are pretty flat. Most of the bones of your face are formed through intramembranous ossification. So you go from mesenchyme to bone versus mesenchyme to cartilage, then to bone. There's a few more steps at a time. So we talk about direction of growth, appositional versus interstitial. We talk about types of ossification, intramembranous versus endochondral. Bone, you are killing living tissue. It's just like getting a cut on your skin. Tissue will die. And so that tissue has to be replaced. So if you have a fracture, yes, you've broken calcium phosphate, but you've also damaged cells, you've damaged blood vessels, you've damaged nerves. All that stuff needs to be a fracture to be healed. So you form what's called a hematoma. It's basically a clot. What you eventually want to do is obviously stop the bleeding. So you clot there, and you start forming more and more bone. Osteoblasts will reach this area and start laying down bone. And you'll eventually form a huge bulge of bone in that region. That's called a callus. Osteoblasts are overly active. They're putting more bone than needed because you have this gap. You have this fractured bone. And you form a calloused area. Just like you have calluses on your skin because you have lots of activity on your hands, with lots of activity here to help heal the area. We don't start. We want to fill in that gap. Now that we've had osteoclast, so osteoclast will resorb bone and get it back to a fairly normal shape. All way that bone is healed. I'm skipping a bunch of parts here. As much as I'm good at testing. A few more conditions that you've probably heard of before. As we age, we tend to lose bone. The reason is because it has to do with hormones and also the activity of those osteoblasts. We're all hopefully not losing bone at our current ages. If we're not losing bone, that means osteoblasts are working as hard as osteoclasts. They're balancing each other out. We're adding bone at the same rate that we're removing bone. That's normal for most adults. As we age, osteoclasts start working more than osteoblasts. The osteoblasts aren't as functional, which means you start losing more bone from your more calcium phosphate in your bones. The term for low bone density, where you get more porous, that's osteopenia. If you have extremely low bone density, that's osteoporosis. You have pores in your bone. Osteoporosis. You tend to see pores in your bone. Osteoporosis. You tend to see this in older women, older females, because there is some role of estrogen, and if you hit menopause, then if you put stress bones, then it tells your osteoblasts that you need to keep those strong. Just like if you constantly use your hands, you're going to have calluses on your hands. If you constantly use your bones, you're going to build up your bone support. When they say drink milk to help your bones get stronger, will that help with osteoporosis? To an extent. Is osteoporosis the only way to get kyphosis? No. You can kyphosis. We're going to talk about this later. That hunchback posture. You can get that from being insecure. You can get that just from. I see a bunch of tall people who are insecure, so they kind of hunch to be more.
It's true. It's true.
The weak muscles. There's other reasons, but that's just one. Okay. I showed you this picture before. You probably didn't know what you were looking at before. But here you can see a fetus. This is the cartilage. This is not yet calcified. So what you see as a skeleton is actually cartilage. That will get replaced over time with bone. Here you can see through. You can see the brain form, and here you can see through the skull because it's just what type of tissue? For a flat bone. It's not cartilage. You can see through it. Sorry? Mesenchyme. We have mesenchyme here, and that mesenchyme will get ossified through intramembranous ossification into bone. Take a moment, please. All right, counts on 3. 1, 2, 3. On 3, say letter. 1, 2, 3.
A.
A, yeah. Cartilage, when we describe cartilage growth and bone growth, it's similar terms. If we want to grow wider, so we're adding cartilage from the outside and removing cartilage from the inside. That's also called appositional growth. Other question?
Just like today all the time. So it's class. And then class.
Reach hand a few more times. Hands on 3. 1, 2, 3. 3, say letter. 1, 2, 3. B, yeah, osteoclasts. Osteoclasts are what remove or resorb calcium phosphate. That gets delivered into the bloodstream. One more. You guys ready a few more time? That's on 3. 1, 2, 3. Find someone with a different answer. I see a mix of answers. Find someone with a different answer. Go ahead. 1 minute. Talk to neighbors. What do y'all say?
Let's see. Yes, the last one should be wrong. Whoever has B.
Sure. Lab check. So I'll point out bonus out to you later on. Now I want you to work on these lab checks. I am going to give you feedback to your pages. I'll hand it back to you. And give these back to you. Let me know if you have questions. But you will need to reproduce this. We have blank sheets of paper, we have markers, you can take a class. So once I give these back to you, let me know if you have more questions. But you will need to reproduce this. We have blank sheets of paper, we have markers and colored pencils. So I will ask you, all right, don't worry.
You guys say it first?